Thank you everyone for coming to the webinar for Vanessa Chin, who's going to be talking to us about her inspirational grant that she received, which was a co-funded grant between ALK Positive Australia and TOGA. It's the first grant we've offered of this kind, so we're really excited to have Vanessa here to talk to us. First, though, I would like to acknowledge and pay our respect to the traditional owners as the original custodians of Australia's lands and waters, and acknowledge their unique ability to care for the country and their deep spiritual connection to it. We honour their elders past, present, whose knowledge and wisdom has ensured continued culture and traditional practices. I'm on the Dja Dja Wurrung country here in Melbourne. So I'll introduce Vanessa. Vanessa is an associate professor. She's a research officer at the Translational Genomics Lab at the Garvan Institute. She's also a medical oncologist at St Vincent's Hospital in Sydney, and she's a conjoint associate professor at UNSW as well. And as I mentioned earlier, she is the inaugural recipient of the first ALK Positive and TOGA grant that was offered, inspirational grant that was offered. And her topic was unraveling genomic diversity in ALK positive non-small cell lung cancer. So I'm going to hand over to Vanessa now to get her presentation up and run us through the really interesting research that she's doing. So Vanessa, if you could put your presentation up for us.
Okay, thank you, John. Let me just make, is that in presenter mode now?
It is in presenter mode.
Okay, well, thank you for having me tonight, and thank you for taking interest in this project, which has been something that I've been trying to get off the ground for some years, and it seems like everything is finally falling into place. So what we're trying to do is look at how we can utilize circulating tumour cells in the blood at a single cell level to improve the management of ALK positive lung cancer. So John has introduced me. This is a little bit more about me. I was a medical doctor before I became a scientist. I got my credentials in 2013. Finished my PhD at Garvan at 2017, and I have been a clinician at the Kinghorn Cancer Centre in Sydney, which is part of St Vincent's Hospital, since 2016. I started my postdoctoral work in 2017, and I work 50-50, meaning that, 2.5 days a week I'm a clinician and 2.5 days a week I'm a scientist. The way things are set up at Garvan is that the Kinghorn Cancer Centre and the Garvan share the same building. So the lower floors are clinical and the upper floors are research, and the idea being that we combine research and clinical medicine together. So it's a really great place to work. So I work in the POWL lab. We are a single cell genomics lab. We look at cancer, we look at cardiac immunology and gastroenterology conditions as well. And we're made up of clinician scientists and bioinformaticians, and also increasingly working in the commercial development space as well. That's a small fraction of our lab. We're about 30 strong. So for those of you who don't know what single cell genomics is, this is just a quick sort of crash course. The more traditional methods for understanding the genetics or the genes of cancer cells use a sort of bulk method where we take all the cells in a biopsy, we mash them up, we extract the DNA from all of those cells, and we run them through the sequencer. And we know that not all those cells are the same. We know that not all of those cells are cancer cells. Some of them are going to be immune cells or support cells or scaffolding cells. And so the results we get are a sort of generalization across all of those cell types. Single cell sequencing or single cell genomics is about trying to understand each of those cells individually. So not just the cancer cells, but all the cells that are in and around the cancer, which are important. And where this becomes important in a disease like ALK positive lung cancer is that we can understand the differences between the cancer cells, because we know that they're not all the same. So this is sort of an overview of all the things that I do. I do some general research, so biobanking is a big part of my work. I do lots of research into lung cancer and into head and neck cancer, and increasingly looking at circulating tumour cells, which is the tumour cells that circulate in our bloodstream, and how we can harness these to run tests and how we can use these in the clinical sphere. So I don't really need to talk too much about this to this audience. This will all be very familiar to you. In Australia, there's about 12,000 new lung cancer diagnoses per year. About 60% of these have now a druggable mutation. We know that the cost of targeted therapies is extremely high and predicted to increase, and that ALK gene fusions are seen in about 4%. Although these TKIs are highly effective, resistance is inevitable. And what we think happens with time is that we develop disease or treatment resistance, so that the cancer clones become more diverse, they become more clever, and they can circumvent the blockade that we're offered with the TKI. And so what we're trying to do in this project is understand how the cancer cells differ from each other and how we can measure that difference and how we can harness that information to impact on treatment decisions. So the overarching aims of this project is to understand how heterogeneity, or how cancer cells are different, and how this relates to treatment response, prognosis, importantly, treatment resistance, and increasingly, how would we use this in adjuvant therapies, so people who've had surgery, and also understanding novel therapeutic targets. So if you look over on this picture on the right, and these are little pictures of cancer cells under the microscope, but if you look at B, this might be indicative of what an ALK positive lung cancer looks prior to treatment. So all of the light pink cells may have a standard ALK fusion, whereas the blue cell and the red cell may have that same fusion but an additional mutation which we cannot detect using current methods of sequencing. And that after a period of blockade with a TKI, with time, those slightly different cells may be the cells that grow up into resistant cancer. So how could we detect this? How could we detect it at diagnosis? How could we detect it at resistance? And how can we use that information? So this is something that we've had in mind for a long time, but just could never quite get the technology right. So a few years ago, I went to a talk and I learned about this machine called a RareSight machine, where we can take a vial of blood here on the left, we spin it all down, and we can extract all of the cells in the blood that are not red blood cells. So all the white blood cells and cancer cells are here in this middle band, and those cells are smeared across a little slide. They're then put into a machine and they're stained. And if you can see this little picture over on the right, there's lots of blue dots, green dots, and one big red dot. And the red dot is a cancer cell. Okay, so the machine can actually detect a single cancer cell in this whole sort of series of immune cells. And in fact, in a single vial of blood, they can pick up just 5 circulating cancer cells. So the power of detection is very high. What then can happen is this little needle comes down onto the slide and it targets those red cells, and it can actually wobble that cancer cell off the slide and put it into an Eppendorf, a little plastic tube, so that we can run experiments on it. And this is a picture looking down this tube, so looking down a bird's eye view, and this is a single cancer cell just sitting in a well. So the fact that you can actually identify a cancer cell in a vial of 10 mils of blood is amazing. And the fact that we can then pick it up with a needle and put it into a well is even more amazing. What we know with ALK positive cancer is that to understand disease resistance or treatment resistance, we need to look at both the DNA, which is the genetic material in the nucleus of a cell, but also the RNA, which is the genetic material which is outside of the nucleus. So these are different. They have different functions and they have a different meaning when we're looking at treatment resistance. So what we now have in the lab is an ability to extract both the RNA and the DNA from the same cell. So this is incredibly difficult. This is novel. This is brand new. So what we can do now is that we can dissolve the outside of the cell, take out the RNA, then dissolve the nuclear wall and take out the DNA, which means we can sequence the DNA and the RNA from one single cell. And what we have been doing and are planning to do is to sequence people at diagnosis, but also people after they've had a TKI and developed treatment resistance. So this is a blown up version of what I showed you before. This is a bird's eye view looking down that little plastic tube where there is a single cell, which is bright green. We then used our fancy kit where we wanted to look at DNA and RNA from that single cell in that single Eppendorf. And on our first go, we were able to extract RNA. And this is a plot showing us that we've got RNA in this little tiny hill in the middle here. And we also got DNA from the same cell. So this was really exciting. And I think maybe the last time I spoke to John, that was as far as we'd got. And a few weeks later, we got this sequencing report. Now, you won't be able to read any of this, but if you go down to this blue square on the left, we were able to detect an ALK fusion. So this cell was a cell line. We knew that it had an ALK fusion because it's a commercially available cell line. And we knew what kind of fusion we were expecting. And on our first go, we detected that ALK fusion. So this is super exciting. This data is only a few weeks old, and we are moving forward with this. So at the moment, we now have about 5 patients. They range from being patients who've been recently diagnosed to patients who have had treatment for some years and are progressing on to their second line of treatment. We've gotten through that process with them. So we've had the vial of blood, it's been spun down, the cancer cells have been identified. And at the moment, we now have them in the freezer and we are waiting to process them through the sequencing pipeline. And what we want to do is process up to 20 cells per patient to understand whether those cells look different. So whether I'm sure we're going to find an ALK fusion in all of those 20 cancer cells, but there will be additional mutations, is what we're hypothesizing, will be found in some cells and not others. And so we can understand what kind of spectrum patients have at diagnosis of different mutations and what spectrum they have at treatment resistance. So what we'd really like to understand is how we can better tailor therapies for patients. For example, if you have a patient who has cancer cells that are very different, so there are lots of different mutations, lots of different cancer clones, is that a patient that would benefit from an additional treatment like chemotherapy, for example, in addition to their TKI? If they have a certain known resistance mutation at diagnosis, would that help us decide between alectinib and lorlatinib, for example? Is this a test that we could use to monitor treatment response? It's definitely very sensitive. We know that. We can pick up, we can pick up 5 cells in a 10 ml vial of blood. Would this test be better than CT scans? Is this going to help us prescribe therapy after surgery? So there are so many potential applications of this work. And we're hoping within the next maybe month, we will have our first sequencing run of the first 5 patients. So this is the team. Obviously, that's me. This is Danelle Yeo. He works at the Centenary Institute, and he's the person that does the rare sites, so the identification. Michael Krasovitsky is another medical oncologist, as is Mal Itchens. I'm sure you'll know Mal, and Joseph Powell, who's my boss and the head of the single cell genomics lab at Garvan. I think that's it. Yes, is everybody there? I think everyone's there.
Yes,
Good. It's kind of eerie talking into the void. So yes, I can hear you and very happy to answer any questions at all. So hit me with it.
I can already see some hands up, but I'm going to just quickly jump in first. What is the difference between the RNA data that you get and the DNA data? Is there some extra benefit you get from understanding the RNA data as well?
So RNA is only a single gene length. So DNA is a long strand which contains all the genes, and RNA is generally a single gene. Therefore, RNA is harder to pick up things like point mutations. So, you know, if a gene is supposed to be A, B, C, D, E, F, I know that they're the wrong letters, but for argument's sake, it's B, A, C, D, E, F. That is much easier to pick up on DNA than it is for RNA. And we know that those point mutations do exist in treatment resistance. But for the fusions, so the ALK fusion, that's an RNA test, okay? So that's the fusion of two genes together. So if we just did DNA, we would miss all the fusions. If we just did RNA, we would miss all the point mutations, which is why we need to do both.
Excellent. Well, I'll hand over to Amanda to check in the chat for us and see if there's any questions in the chat, and then we'll go to the people with hands. Amanda?
I think we're just jumping to the hands, John. We'll go to Ellie Bolton first.
Sorry, just trying
That's
to
okay.
check out technology, and my dog is barking. Vanessa, I think this is really interesting because you can be so specific in looking at the one cell and look at those mutations really easily.
Mm-hmm.
So you know that a few of us on here have been on the ALK trial. Just hang on a sec. Radio. So we kind of want to understand how things look in terms of mutations over time and with progression.
Yes.
One of the questions I have is what happens if you have, for instance, something like metastases only in the brain,
Mm-hmm.
And there isn't a large load of cancer cells in the blood. How do, because you can get one cell out of the blood, do you have to take larger amounts of blood, or how do you manage that scenario?
Excellent question, Ali. And I don't think we really know. So we know with circulating tumor DNA, which you may be sort of more familiar with because that is a sort of a more standard test. We know that your ability to detect ctDNA is directly proportional to how much disease there is in the body, but also how much blood you take. Now, circulating tumor cells, I think, is probably going to be a little bit more sensitive than that because ctDNA has its limitations, and you know we can discuss that if anyone's really interested. But ctDNA, you need to know what you're looking for before you can find it. Whereas circulating tumor cells, because the cancer cells look so different from the immune cells, we're much more likely to detect them. Now, in the case of brain metastases, by virtue of the fact that there are brain metastases, there must be some tumor cells circulating in the body. What that threshold is going to be, I think we're only going to find out by sequencing more patients and understanding the correlations between the scans and what we can see and then what we get back on the blood. At the moment, we're taking 1 10 ml vial of blood, and that's come down to money. But I think when we get these preliminary results and indeed with the funding that you've given me, progress to probably a clinical study where we may be using the Dynamark network or the Aurora sort of framework, we will have capacity to take more. So I think the short answer is I don't know. I think it's going to be a bit more sensitive than ctDNA though, because we're not relying on a mutation being something that we understand. We can just look for the cell.
Okay, thank you, Vanessa. I look forward to the next, to the rest of the work rolling out.
Thank you.
All right, we'll go over to Lisa Briggs now.
Thanks for that, Vanessa. That was awesome. I don't know if I missed it at the start. Were these just metastatic ALK Positive Lung Cancers.
Yes.
Yeah, okay. And are you able to detect the variants 1, 2, and 3 when you do your RNA-seq, or is that?
Yeah, so basically we're running this through a standard Thermo Fisher Oncomine test. So we will be able to detect sort of standard fusions and novel fusions. So it's actually even a lot more powerful than I was expecting. So our first run was amazing. The data was very clean. What we were worried about was, because we've only got one copy of the DNA, we've got a single cell, there's only, one DNA pair. So we have to amplify that DNA up using polymerase chain reaction, where you basically, it's like running it through a photocopier, right? And we know that when we do that, sometimes the quality of the DNA and the RNA goes down, but it was fine. It worked really well. And we were able to detect the fusions and because these were cell lines, we actually had comprehensive sequencing done already. So we knew what we were looking for, and we were able to find all the things that we knew were there. So I don't know if this is just beginner's luck, but the short answer to your question is yes, I think we'll be able to detect all fusions and novel fusions.
That's brilliant. And just on Ali's question, is it possible with CSF?
Yes, I think it would be possible. I think I can't see any reason why it would not be possible. The only thing is we need to be able to differentiate between the background cell types and the cancer cells, but CSF should technically have no cells, so that would even be easier.
Because I think one of the barriers that we seem to come across with researchers at the moment is the hesitation in putting patients under, doing a spinal tap is, a lumbar puncture, I think does carry some risk. And I think a lot of patients who have had brain metastases sort of would put their hand up and say, yes, but I'd be happily do it. It's just about finding the right people to say yes. And so I just wonder whether research will go down that route at some point to enable more opportunity to get those answers that we're all looking for.
I mean, I think it's funny, isn't it? Because lumbar punctures are really out of fashion, whereas when I was training, before I did my oncology training, we did them all the time. I personally think it would be, in a motivated patient who's prepared to undergo a procedure, which in fact isn't that painful. I mean, they really go on about it on TV, don't they? Like anytime you see someone having a lumbar puncture on TV, it's terrible, but in reality, it's not so bad.
Well, Vanessa,
yes,
I have worked previously as a midwife, and women are having spinal anaesthetic a lot.
Yeah.
Quite a lot, and are very accepting of it, so
yes,
it's a completely different realm of health. But,
it's funny, isn't it? It's just, and in neurology, they do them, all the time. But for some reason in cancer, as I said, they've just sort of gone out of fashion. But I think what would be fascinating to know is the blood going to be sufficient here? Like if someone's got brain metastases, are we going to be able to detect it in the blood? And that's probably the answer we'll get first. And if it isn't, then moving into CSF. But would be really interesting, Lisa, to see what the ethics, like the governance office would say about that. I don't know, because they give me a hard time with the stuff I'm already doing. So, yeah.
And
do you think it would hold? Oh, sorry, Liz.
With brain metastasis, it's not even a question brain tumours, it's not even a question. If you have a primary brain tumour and they need to access information without surgery, it will happen. But for some reason, because it's a metastasis in lung, there's resistance. So I think a lot of us would be pushing for this. And maybe that's something that ALK positive community can maybe put a grant out for at some point.
Yeah, like the management of brain metastases.
Yeah.
And I think helping with those protocol designs and ethics applications, I mean, the last RGO ethics application I put in for the first time asked me, had I had a consumer review my protocol? And I think that's a sort of powerful way of saying, well, yes, I have, and they tell me that they're prepared to have this invasive test done. And I think the RGO would listen to that.
Yeah, thanks.
All right, we might move to John Cannings.
Hi, Vanessa. Thank you very much for a great presentation. My question is more about enrolment. You've got 5 patients. For those who are already on TKIs who may progress, how are you going about enrolling or getting the word out?
So at the moment, we're a little bit constrained by time. So at the moment, we have to take the blood and we have to process it within 2 hours. So it's being done just on the St Vincent's campus, and we're using the machine which is over at the Royal Prince Alfred campus, which is just where the Centenary Institute is. So we've already been talking about this, myself and Melinda Itchens, about how we could expand the catchment. And what we'll probably do is try and open it at Royal North Shore Hospital where she is, and at Royal Prince Alfred Hospital as well, because that could still get us within that 2-hour time frame. If we can show what I think we're going to show, which is that this is going to be a helpful, useful test, then what we're going to need is more rare site machines. If we had one centrally at Garvan, then it would make it easier for us to run things in real time and expand it out to sort of more metropolitan Sydney. We're working on a few methods. I sort of had something on the last slide about fixed cells versus fresh cells. So cancer cells that are fresh, meaning that we haven't put any chemical fixatives on them, are much easier to work with. The RNA is much easier to work with. Fixatives tend to be sort of methanol, ethanol based, and they make the cells easy to work with from a handling point of view because they're stuck to the slide. You don't have to refrigerate them. You know, they kind of stand still in space, but the RNA degrades very quickly. Now, on our first run, we may be able to actually use fixed cells, which is not something that we had thought we'd be able to do. If we can use fixed cells, then this sort of blows out of the water. So that sort of 2-hour time period that we're working with now becomes a 96-hour window. And that would enable you to enroll even outside of New South Wales and kind of courier your blood in overnight. So I think that's more of a medium-term plan. I think the first step is going to be widening our catchment to those 2 neighbouring hospitals to the machine, which is at Centenary, and then, you know, probably purchasing another machine. And then if we can work with the fixed cells, that's what we'll do.
Is that all from you, John?
Yes, brilliant. Thank you very much.
Excellent. We'll go now to Peter Spolk.
Oh, did he press the wrong button maybe?
Fantastic presentation, and a new concept to me. Maybe I've missed something, but I want to learn more about the variety of novel forms and how many cells you would need to assess to be able to say, I have a handle on where the critical issues are.
Really good question, Peter. Again, it's an unknown. Where I think it's going to be easier is in patients who've progressed on therapy, because we know from bulk sequencing techniques that they are very likely to have resistance mutations. Therefore, they are much more likely to have a variety of different cancer cells making up their cancer. So that rather than having 99.9% of one clone and 0.1% of another, they're probably going to have a mix that we can detect. And this is then going to just come down to money and what we can find. So in someone who's got a high disease burden, we may get 100 cells back, and it would be great to do all 100 cells. In someone who's got a low disease burden, then we may only get 20 cells back. So in patients who are newly diagnosed and treatment naive, I think we're going to have to sequence a lot more cells. Maybe it's going to be 100, maybe it's going to be 150. So maybe, if we're talking pie in the sky and money's not a problem, we might need to analyze 30 mils of blood, 40 mils of blood, and then sequence all the cells we get back.
Great.
So yes, for any statisticians, it's a nightmare, but I think we're still on the, you know, on the learning curve.
Can I sneak in a second question? And that is, when you've got this profile, if you like, of novel fusions, etc., is it going to be clear what the next treatment ought to be? In other words, how well integrated is our diagnostic information with effective treatments?
So at the moment, we use it not at all, especially when we're prescribing the first therapy. And that's because, you know, we see an ALK fusion, it's a dominant fusion, we don't really get any other information, especially because it's all done on biopsies, it's all done on those bulk techniques that I showed you right at the beginning. Any one of you who's had ctDNA done during the course of your treatment, you'll have a much more informative report because you do get that sense of heterogeneity. And so I think we are using that sort of more granular data now for second and subsequent line therapy. What we're not doing is using it up front. And in your community, you'll maybe know people who've been on crizotinib for 10 years. You may know someone who's been on lorlatinib for 6 months. So there's this massive spectrum of people. Clearly that person who's been on crizotinib for 10 years has a very bland tumor. You know, they've got no heterogeneity. They've just been using the same target for 10 years. And that person on lorlatinib for 6 months had a very heterogeneous or different tumor that just could not say suppressed. So in the EGFR space, we know now that adding chemotherapy to a TKI helps people stay under control for longer. But we know instinctively that probably not everybody needs it. So I think that's going to apply in the ALK space as well. I mean, these trials are coming through now. But at diagnosis, a fundamental question might just be, does this patient also need chemotherapy? I mean, that could be something that we could solve with this technology straight away. And then the harder question about, you know, really with confidence saying, this patient only really needs a first generation TKI, whereas this other patient needs a third generation. I think that's coming later.
Thank you very much, and let's celebrate blandness in a way that
yes.
Thanks, Peter. It's just thrilling. I'm just so chuffed to be here. John, you've got a question.
Yeah, I thought I should put up my hand and join the queue with everyone else. I did see there's a couple of questions in the chat, Amanda, if you could open up the chat and look at those ones too. This sounds like, to me, this is that first step in what, you know, has been put out there as personalised treatment, and the dream of being able to actually choose the right approach based on the knowledge of your cancer. To be able to do that, obviously, talks about access that you were talking about. You know, you're limited to this little pool around you at the moment. Are there more of these machines in other states, or, you know, how many of these machines are in Australia? And
yeah, so there are 2 in New South Wales. There's one at the Melanoma Institute and one at Centenary. I don't know about other states, John. That's a really good question. I haven't heard, but they seem to have everything. It would not surprise me if they have one.
I think Stephen would have one, Vanessa.
Really?
Yeah, I think so. I'm going to ask him.
I'll talk to him about it. But like anything, John, once they become useful, then everyone will buy one. So I think, you know, the first step is for us is to show that it works and that it has a clinical application. And if we want to be a little bit sort of cynical here, once the company realizes there are clinical applications, they will not be able to sell them fast enough. So I think that's why this, and what we're maybe aiming to do is try and get a paper out quite quickly, really a methods paper about how we've done it, what the results are, to sort of start getting, putting a notch in our belt to get us that next leg up for next funding, but also sort of advertise the 2 technologies that we're using. So we've talked a lot about the rare site, but the technology where we can extract the DNA and the RNA is also very, very novel. To give you an idea, the company said to us, your first test kit is going to be $30,000. And we said, well, we're not paying that. come on, we're the Garvan Institute, please give us a free kit, because often they do. And they said, sure, we'll give you a free kit, but shipping will cost $10,000. So our free kit was 10 grand. There you go.
So that goes to my follow-on question, which you mentioned that the 5 patients which the grant has funded to help you
Yes,
do
yes.
are already there and taken for you. If there was other patients who wanted to access the same type of information, do you have an idea of what that would cost out of pocket for someone who wanted that information but couldn't be part of
Yeah, so
trial?
I don't know that we would be able to provide a service like that now. Anyone who's coming through the sites that are approved for the study, we could probably, work something out. But, you'd have to be a patient at the moment. Today, you'd have to be a patient at St Vincent's. Potentially in 6 months, you would have to be a patient at Royal North Shore or Royal Prince Alfred. Giving results out like this outside of a clinical research field is hard and contravenes a lot of our ethics. So I think what we'll probably do, John, is if this all works the way that we're hoping it'll work, Melinda and I will probably write up something quite quickly so that we can then have a bit more freedom. And then the only barrier is going to be money. I mean, the only barrier is going to be money. But it means then if people want to do it, we can try and make it happen. Yeah.
Because circulating DNA tests are cheap on their own anyway, and people are happy to, or at least some people are happy to try and cover that expense themselves to get that additional information. So there could be opportunities there. But I'll throw to Amanda to see if there's any questions that were asked in the chat that we need to address.
For the most part, they've been answered. There was one question from Shamila who had prior to this little part of the conversation suggested that she'd be keen to enrol. She's got a rare and strange mutation, but asked and answered. So that information's there. Yeah. Other than that, it's more conversation than question. So everything's been covered.
Well, I'll throw it out for one last opportunity if there's any other last questions. Otherwise, I will say thank you very much to Vanessa. It's greatly appreciated you giving up your time outside of your office hours. We know how busy all the doctors are. We greatly appreciate it. You took what was quite a complex topic, and I think you explained it in a way that was really understandable to us as patients. So thank you so much for doing that. It's a really thrilling bit of research that you're doing, and I'm so glad that ALK Positive Australia was able to help get this up and running. And as I said, hopefully this is just that little bit of boost that can take you to that next level and demonstrate application and attract greater funding in the future to make this far more available to other patients. I'm really pleased that this was our inaugural grant and it went to you. And we really look forward to having you back, in a little bit of time, maybe after you've got some results that you can publish and talk us through what you learn. Thank you so much.
I would love to do that. Thank you so much for having me. And look, it's really nice talking to people who are kind of the direct beneficiaries of the research. And when it comes to that next stage of writing the protocol and applying for more grants, then John, I'll maybe be knocking on everyone's door to see who would like to help support it, because I think that's what makes the difference now. We need that that support. So that would be amazing.
We're here for that reason.
Happy to help, Vanessa.
Yeah.
Thanks, everybody. Thank you.
Thanks for explaining it so well.
Yeah.
Not my pleasure. I can talk single cell genomics all night. So John gave me a limit. I stuck to my limit.
Really, really useful. Like I said, totally understandable as well as it could be anyway. So thank you so much for your time. We really appreciate you coming and being part of our team. I'll say good night to everyone. Thank you very much.
Good night.
Thank you, Vanessa.
Thanks. Bye.
Bye.